Welcome to JAX Tech Talk, formerly Mice Tech Talk, with the technical information scientist of the Jackson Laboratory. My name is Caitlin, and my role as a technical information scientist is to serve the research community with education and practical solutions. Some long-time viewers might notice that we recently changed our name to JAX Tech Talk, and I wanted to assure you that we're the same people and the same great show in a 15-minute format. But our new name really reflects future thought-provoking topics and practical solution-based content. So for any of you that are watching live, use the comment section of the video to tell us where you're viewing from and any related questions that you might have today, and we'll try to answer those. And I already see that the lab scientist said hi, so I give you a shout out there. So today, let's talk JAX Swiss Outbred mice. Today, I have my colleague here, Kelly Brackett, on the show with me, and thanks for joining us. So why don't you tell us a little bit about what your role is at JAX?
Sure. So my title here at JAX is Scientific Advisor, and in my role, I sort of split my time between teaching mouse biology and genetics to our animal care staff and helping to manage and advise our mouse colonies. Specifically, it seems that my focus tends to be on our outbred colonies, so that's why I'm here today, I guess.
All right, so since your main area of expertise is those outbred mice, let's talk about those JAX Swiss Outbred mice, also known as JRX. And the stock number for those of you that are viewing, in case you want to look it up, is 034608. And these mice are relatively new to JAX. So could you tell us a little bit about the strain origin and background, kind of where they came from?
Sure. So we actually just received breeders in 2020, so just in this past year, and we got them from the Animal Resources Center, or ARC, A-R-C. That's why we have that nomenclature. And this research facility is in Canning Vale in Western Australia. So they had a long journey to get to here, us on the east coast of the United States, 2 continents and the Pacific Ocean. So they've traveled a long way. These mice are really similar to CD1s in that they were derived from the same common ancestor. So if you've used CD1s in the past, you can expect the same coat color, large body sizes, and similar breeding statistics such as really high fecundity. However, those 2 colonies, what we have now at JAX and the CD1s, have been separated since the early 1980s. So there is some potential for genetic drift between those 2 colonies.
Awesome. Now, you did say high fecundity in this strain. So do you know how many mice per litter that we typically see?
It's a lot.
Yeah.
So far, what we've seen is like 14 or 15 on average. So
wow.
It's an insane amount of pups per litter.
Right, because a black 6 is what, 6 or 7 pups
yes.
on average? Yeah, so almost double in size. Wow.
Yep.
Okay. Just a reminder to everyone, go ahead and submit your questions now so that we can answer them before the show ends. I do see we've got some viewers from Saudi Arabia and Texas, so welcome. Thanks for joining us. So to get back to the JRX mice, this is an outbred strain, so this means that they don't follow the traditional brother-sister mating scheme. So can you tell us how we breed these mice at JAX and how we do so that they're still considered to be outbred?
Sure. So to be considered outbred, all you have to do is do non-brother-sister mating. So just males and females of that colony from different pens. But we use a very specific breeding scheme called the Poiley method. And this is essentially like a rotational breeding scheme. So in this method, and the way we do it here at JAX, is that the colony is actually separated into 32 different quote-unquote lines. And every generation, the line that is mated to each female is rotated. So for example, if in generation 1, line 1 female is mated to line 2 male, then in the next generation, that line 1 female, the female pups that come from that first litter, are going to be mated to line 3 male, and so on and so forth until you go through all 32 lines. This really helps us keep track of the relatedness between mice and ensures that any breeding pair that we put together are as distantly related to each other as possible, which helps us to maintain genetic diversity of the population as it ages.
Awesome. So that made me think of a question that we get frequently in technical information. So we also have the JDO mice at JAX, the stock number 009376, and they're both kind of named similarly. They have that J semicolon. So are these two strains similar in any way, or are they just completely different mice?
They're similar in that they're outbreds or random breds, but they are very different otherwise. So the JRX are derived from a single stock of mice, and that's why all of these mice are going to be albino if you get JRX. But the JDOs, or the diversity outbred, are actually derived from 8 different inbred strains, all mixed together, all with highly diverse alleles and phenotypes. And typically, if you get a cohort of JDO, if you order a cohort of JDO from us, you're going to see a lot of different coat colors in a single box, and that's because of all these different founders mixing together. The JDOs are actually more, I'd say, more genetically tractable as well, because with each of those different 8 founders, we know every distinguishing SNP, single nucleotide polymorphism, across the entire genome of each of those 8 founders. So if in a particular mouse you're questioning, what is the particular allele that's causing this phenotype, you can dive into that very easily using the JDOs. The same is not true of the JRX. We don't really have a whole lot of information about what types of SNPs that they have. We just know that they are generally outbred Swiss mice. And in terms of genetic diversity, the JDOs, because they come from a variety of different inbreds, and because of the mating scheme that created them, they have really high heterozygosity, meaning that you have heterozygous loci across the genome. So about 85% of the loci that we can genotype using things like gigamuga will be heterozygous. So 85% is really, really high. And we are in the process of doing a similar characterization for JRX, but we totally expect that their heterozygosity is going to be somewhere around 20 to 30%. So while they are more diverse for sure than any inbred strain that you would ever encounter, because those are 0, the JDO are definitely a much more allelically diverse population. So we're working on doing the sequencing and getting those numbers, and we expect those results sometime in the late fall, and we will update customers with those numbers that we find in the strain data sheets online.
Awesome. Yeah, that's a great description of how those strains are really mostly different. The only similarity being that they're both considered to be outbred strains. Yeah. We've also been getting a lot of requests about these mice, and we do have a colony that's currently sized to accommodate orders of 100 or more at a time. But do you know how customers might be using this strain?
So they can be used for a variety of different reasons, and probably reasons that we haven't even thought of yet. But this stock is mostly used for pharmacology and toxicology studies. Because their level of genetic diversity can help with discovering a diversity of reactions to different compounds that you might want to be testing and doing translational research with. They more closely represent the diversity of reactions that could possibly happen in humans, much more so that an inbred strain could ever tell you. They also, interestingly, even though they're diverse, have fairly consistent body weight. So it makes it much easier for doing dosing studies for people that are working in this type of research field. Customers have also used this stock as sentinels in their animal health programs, really because they're inexpensive and they're easy to breed, as we talked about before. And also because of their excellent breeding characteristics, they have also been used as foster moms or for embryo transfers. They're really, like an all-purpose laboratory mouse. One interesting thing, though, is we've begun the characterization of the colony that we have at JAX now, is that we see that they respond really well to STZ treatment, which is STZ is a chemical that basically induces diabetes. And these mice respond really quickly and effectively. They have very high blood sugar levels, after a week of treatment.
Yeah, I'm really excited to see that data come out, being the metabolics expert in our technical information group. So I can't wait to see that data, and that'll also be posted on the web as soon as we finished it. So I do see one question. Can you talk about the health status of these mice?
Yeah, sure. So we have colonies of the JRX on both our East and West Coast facilities in Bar Harbor and Sacramento. And we actually are keeping these colonies at our maximum barrier level. So our highest level of pathogen-free health status. So basically wherever they go, from our facility, there's no quarantine that's going to be needed for these mice when a customer receives them. They're going to be of the highest health quality that we can provide, and I think it's very beneficial, especially for people that are doing pharmacology and toxicology studies, to know that the mice that they're receiving are of the best health status possible.
Right. Yeah, and that'll save researchers a lot of time not having to worry about quarantining and such. I do want to point out we do have a few links either above the video or below the video, depending on which, if you're looking at YouTube or LinkedIn. But we've listed the strain data sheet for the JRX mice, as well as 2 publications that relate to the JRX mice for you to check out. This is all the time we have for today. So thank you for joining us this week. Our next JAX Tech Talk is called Let's Talk Breeding Services, which will be live on Tuesday, July 13th. So follow JAX Tech Talk on LinkedIn so that you get notified anytime we have a new episode, and we'll also be posting useful links there. Or you can subscribe to our playlist on YouTube. So this is Caitlin saying, stay healthy, stay safe, and stay excited about research. Bye, everyone.